Okay, today's webinar is on advancements in toxicological hazard and risk assessment for food safety, exploring new approach methodologies and predictive models. This webinar has been developed thanks to ILSI Europe. So as a small presentation, my name is Geraldine. I work at ILSI Europe. ILSI is a branch non-for-profit organization. At ILSI, we generate science-based guidance for the food sector to foster collaboration and facilitate consensus building between three bodies: the academia, industry, and the government. These are some of the members that ILSI has, and we invite other members to also join ILSI Europe. So as I mentioned, we are a tripartite model. We generate science-based solutions for the food sector, and we are developing all these solutions towards a more sustainable food supply for all. To have a productive webinar today, I ask you to kindly mute yourself unless you are speaking. If you have a question, you can raise your hand, or also you can use the chat, which is below below Zoom. You have a bar, so you can also use the chat if you have any questions on today's webinar. The speakers of today's session are Cyrille Krull and Professor Cyrille Krull from University of Applied Science Utrecht, and Professor Bas Blaauboer. Professor Matthew Winken and Nienke Kramer are the speakers for the session on May 30, so you are invited to also join that webinar. And with it, I give the floor to Bas, to Professor Bas Blaauboer.
Thank you very much, Geraldine. Good afternoon. My name is Bas Blaauboer. I'm the president of ILSI Europe. I'm an emeritus professor of toxicology at Utrecht University, and I'm trying to coordinate this course that we are now giving. The aim is, as Geraldine explained, to introduce to you the methodologies that are described in this 2016 paper, in which you see the authors of this paper, and as you can see, also Cyrille Krull was one of the authors of this paper. Can I have the next slide, please? I give you just a flavor of what is in this paper. It is a flow diagram that is describing a number of steps that can be taken in the assessment of foods or food products or food matrices or whatever you have. And it is meant to use as much as possible the new approach methodologies to avoid animal testing. And as I say, that is quite different from the prevailing methodologies that are in use at the moment, who is mostly focusing on the apical endpoints in toxicity testing and in animal experiments. Next slide, please. This is the flow sheet. This is just to impress you by showing this in complete, because there are so many steps to be taken in this, and what we would like to do during this course is to show you that what are the essentials of the different steps in the whole process, and how we can come to a more rational risk assessment than the present methodology at the moment. So the next step, please. Most of these steps will be explained during the course, and the aim of the two webinars is to give you a flavor of what is possible during the hands-on training that we are planning doing in the beginning of July in the Brussels offices of ILSE Europe, in which we are going to use this scheme with a number of compounds, hopefully, and see how it works and where the pitfalls are and how we can solve these pitfalls. Can I have this next slide, please, Geraldine? So one of the issues, for instance, is that we could use a number of, let's say, computer-based methodologies that can tell you something about the structure-activity relationship or the structure-property relationships. Hopefully, we can do that in a quantitative way. Other things that are important is can be used in the bioavailability of the compound and the use of, let's say, computer-aided models to describe what's going to happen in tissues, organs, or even in the organism. So next slide, please. The program of today is that after the introduction of the workshop done by me, we will give a presentation on the present methodologies on hazard and hazard assessment. We will do that in two steps. I will give a presentation of the present methodology, and Cyrille will give you an overview of the possibilities that are mentioned in this scheme and how we can use them in the risk assessment of compounds. Yes, please. On the second webinar, which is planned on the 30th of May, we will go into detail where we will explain a number of possibilities to understand much better the mechanisms of action of toxicology. We also will explain the use of kinetic models, computer models that describe the uptake, the distribution, the metabolism, and the excretion of compounds. That will be done by Nienke Kramer. By the way, the presentation of the adverse outcome pathways will be done by Mathieu Vinken. And in the end, we will come back to you with the possibility to describe how to use all these methodologies to come up to an in vivo explanation of the toxicology of the compound and to do a risk assessment on the basis of this. Next slide, please. And what I also will do then is give an introduction on the hands-on training in July. Yes, please. So, well, you were here, so you were registered, I guess. So this slide is not necessary anymore. What's the next one? Okay, this was, oh, this is just for other events. What I will try to do now is share my screen with you and see how this works. Can I do that, Geraldine?
Yes, Bas, you can go to the share screen button because.
Okay.
Yes, you're starting to share.
Ja. Eindelijk, ja. Okay. What I will do now is give you a short presentation on the risk assessment and what is the history of it, what is the possibility to use it in the present situation, and how to use it in the future. So when we are doing a risk assessment, it is always containing 4 different steps in the whole process, whatever you do. If you do it with animal experiments or if you do it in another way, that doesn't matter.
Bas?
Yes?
Yes. We are able to see your screen, but we are not able to see the slides you are sharing.
Oh, that is strange.
So we can see the presentations you have on your folder,
Yeah.
but we don't see the presentation per se.
Oh, I see it on my screen, the full presentation. So
Maybe, but
I
I
will
think,
stop it and start
yeah,
again.
indeed. Please, when you start again, I think you need to choose to read the whole files and not only one single screen. I think you need to choose screen. Okay,
No.
much better.
Yeah, are you you can see it now?
Yes.
Okay, now we go to the presentation mode and go back to the beginning of the slides. You can see it now?
Yes.
Okie
Yes.
dokie. So what I said was we have the 4 different steps in the whole process. The first one is the knowledge that we need to have on the possibility of a compound to interfere with a biological system that will result finally in some adverse action in an organism, for instance. And that's what we call the hazard identification. It's just a quantitative description of what is possible with a certain compound in a biological system. The second step of the whole process is very essential because it describes the dose-response relationship, and that's what we call a hazard characterization. I come back to that in one of the next slides, but it is important to realize that there is a relationship between dose and response. Having said that, it's also logical that we have to talk about exposure assessment, because if the exposure is very low, the dose will be very low, and the response will be different from a higher dose. And bringing all these elements together, exposure and the dose response relationship, it gives you the possibility to characterize the risk in terms of what's happening in a given exposure scenario with a certain compound with regard to the possible adverse effects of a compound. Also have to remind you that we have the exposure on the other sides, and on the other side of the slide you can see the effects. And in between we have the description on how the compound can reach the site of the effect. And that's what we call the kinetics of the compound. The description on how it moves around, how it can be metabolized, etc., etc. I added this slide also because we have to realize that it is not always the compound itself that is doing the effect. It may be the result of a metabolite formation that is also the possibility of a compound to react with the biological system. So a toxicant might be called a toxicant, but it is probably the result of the ultimate toxicant, the target molecule that gives you the effect. So we have a number of questions to be asked to ourselves. The first one is, what is the compound capable of doing? Hazard identification. The second one is, what is the likelihood of such an effect given a certain exposure? And that is the risk assessment, that is the dose-response relationship. And finally, we could be asked to give an advice on how to use a compound safely, and that is the final question. What level of intake would be related to health risk if you consume it daily over a lifetime, for instance? Or if you're talking about acute toxicity, what is the acute dose of a compound that gives this problem? Well, as I said, the present situation with regard to risk assessment is the classical way. Animal experimentation, quantification, trying to find a no effect level. And then we are supposed to also think about the fact that we have a lot of uncertainties, and all these uncertainties need to be taken into account if we are having to extrapolate the compound to, for instance, the human situation. And that's what we do with safety factors. I come back to that later also. And the establishment of the safety standards that are the result of this are then, for instance, expressed as an acceptable daily intake of a or a tolerable daily intake, or whatever you would like to call it. There are lots of uncertainties in this approach. For instance, the fact that you are only looking at the endpoint of the toxicity without understanding the mechanism of toxicity. And we think we can do more, much more, if we take these into account. And that is more or less the aim of this course. As I said, it is important to understand the dose-effect relationship of a compound. And this is, well, my flimsy description of the use of a computer to make such a dose-effect relationship. You see, the line is not so straight as you could expect. It's drawn by hand. And what you see here is that many toxicological processes can be described by taking into account a threshold below which nothing happens, and above which you see an increasing amount of effect coming up. There are situations by which it is by no means clear that there is a threshold, or to put it in other words, to make clear in this figure that we are not sure that we can determine it. And this is, for instance, the case now in the risk assessment for carcinogenic compounds, for instance. I come back to that later as well. There is one other possibility, that you have a dose-response relationship with this, in this shape. So you see here, when the compound is absent at the y-axis, there is a negative effect of the compound, and it goes through the abscess, etc., and then going up again. And here you see a trajectory in which the compound has a beneficiary effect. That is, for instance, the case is foodstuffs that we need to survive, or vitamins, or whatever. You could also say that it is a drug in the situation by which you have a disease situation that helps you overcome this. So how do we get to a no effect level? In an animal experiment, you can see that you can have, let's say a choice of doses and groups of animals, and that gives you the incidence of a certain effect. And these are the measuring points of such an experiment. And you see that one of the reasons that the no effect level approach is not so precise is that, well, it's just a matter of the doses that you choose, where exactly this no effect level is. It might be higher or lower than the point that you have in your experiments. And that's why the no effect approach is now most of the time displaced by the so-called, how do you call this, Cyrille? Help me.
Calculated?
Yeah, the calculation of the benchmark doses approach. That's what I was looking for. Yeah, which is a statistical use of the same database, but gives you a much preciser data number for the risk than only the no effect level. So in the end, this ends up then in what is now the general approach, acceptable daily intake, defined internationally decades ago. No effect level. Also, you can see this is defined decades ago, and that gives you the reason for having safety factors. One of the reasons is that you have an experiment with the uncertainty in it, but you also have the possibility that the difference between an animal and, for instance, the human situation is quite big, and that's why we need to take into account these safety factors only for this reason as well. And this slide gives you that problem more or less. We take a safety factor for the interspecies comparison from animals to humans, but you also have to take into account the fact that the average human is probably less sensitive than more sensitive groups. Think of children of the older age, or diseased people, or whatever you think of. The normal procedure is to take then safety factors of 10 for each of these factors, so you end up with a safety factor of 100. And it must be said, in many cases, these safety factors are serving well the safety of a compound, especially in the food area. Also remind you that these differences between animals and humans, and even the more sensitive humans, are probably related to, in the first place, of course, the uncertainty in the experimental data, as I said earlier, but also in the fact that there are differences in the kinetics of the compound, or the activity of the compound in the human situation as compared to the animal situation. And this also applies to the difference between the average person and the more sensitive person. As I said, in a number of cases, it is very difficult to describe the causal relationship without a threshold, with a threshold, and this is what we call the non-threshold effects. You could say probably that this might be related to interactions with essential and unique processes in the organism, for instance, the DNA, in which, in a hypothetical case, one change in the DNA molecule could lead to a problem. So that would mean that the threshold for such a compound is one molecule. And this is very hypothetical because we have so many safety measures in our body that this is only a hypothetical thing to think about. And what we do take into account is the fact that we really don't know where this is. And the most safe way of taking this into account is to leave out the threshold and have a linear response curve going to the origin of the figure. And then the risk assessment is based on the fact that we only take into account, let's say, the safety of 1 in a million possibilities, etc., rather than 0. To end this general introduction into risk assessment, I have to say that there are also a number of follow-up steps to be taken. And one step to be taken is the step in the risk management. How do we deal with adverse outcome? How do we deal with acceptable daily intakes? How do we cover the possibility that they are surpassed? And that is the area of the risk management. And I also mentioned the fact that there is a very important role for communicating the proper risk, etc. So this ends then my presentation by also showing you the risk management circle. If you have a problem, if you think there is a risk, you look at your options, you take decisions and actions, evaluate what the result of this is, and see whether you improve the situation. So by having said this, this is my general introduction in risk assessment. There may be questions. If you have questions, put them in the chat or raise your hand or whatever. And then I stop here. And what I would like to do now before Cyrille starts her presentation is that we would like to ask a number of questions to you in the audience. Is it okay with you, Cyrille, that we do that now?
Yeah, that's fine.
Okay. So, Geraldine or Hugo, if you are present there, could you take over again? I will stop my sharing of the presentation and ask either of you to start the quiz. And the quiz is to have a number of questions asked to you to get to know you a little bit better, so we could probably also evaluate what you would like to hear. Yes, Geraldine?
Yes, just a second. Hugo will launch the poll, just a second. And now,
Here we go. The poll is
Yes.
launched. I'm still letting the poll run because I'm still waiting for people to vote. Please feel free to vote. Okay, so we have actually 2 minutes since the poll is running. I can see there are still people voting. Okay, so we have actually 28 out of 38 that voted, so we have 50 seconds more. 5 seconds more, and we close the poll. Thank you, everyone. Okay, Geraldine, would you like me to share the poll?
Yeah, please.
Yeah, I will do
Yes.
that.
So what we see here is that we have a nice representation of the different groups. We have people that are having a background in toxicology, right? We had more questions, did we?
Yeah, if you scroll down, you should be able to see the.
Oh yeah.
Yeah, basically it's one poll with 4 questions.
We have people that are having experience in safety assessment, and we also have people that don't. And we have people that are having experiments with cell culture. Okay, so we are pleased to see that we are having the different backgrounds present, and well, we hope that we are giving you the proper information to work on this in terms of the July workshop, but we come back to that later. What I would like to do now is to give the floor to Cyrille.
Yeah, thank you, Bas. Let me see whether I can share my slides. Yes. Can someone of you confirm that you see my slides?
Yes, I see them.
Yes.
Okay. But I don't see my slides. Okay. And is it also on the first slide?
Yes,
With
in
the
the first slide.
introduction of myself?
You have to start the presentation.
In the presentation mode?
Yes, you need to go to. Now it's okay.
Yes, there it is.
Okay, great. Okay, so thank you so much, Bas, for the nice introduction, and also for the participants to know a little bit more of your backgrounds, where you're from and what your experience is. Good to learn that there are people with a toxicological backgrounds and experience in risk assessment. That's very nice. Let me briefly introduce myself. As said, I'm a professor at the University of Applied Sciences in Utrecht. And my research group is about innovative testing in life science and chemistry. So I'm specialized in the development and implementation of animal-free innovations. And previously, I worked for TNO for a very long time. And there I was head of the regulatory toxicology, mainly on in vitro toxicology. And did my PhD here in Utrecht and before in Maastricht. And I'm also the vice or the co-chair of the task force on animals alternatives to animal testing of ILSI. And in that task force, we discuss a couple of things like we do now today. And if you are from the industry and interested to join the group, let me or Geraldine know. We would love to have you in the team. So to briefly summarize what Bas just was mentioning, the scope of the safety evaluation in general is to characterize the toxic effects of a compound, to identify the potential target organs, of course, the dose dependency, and also to find out whether it's reversible or not. That's something Bas touched upon if we are talking about carcinogenic and mutagenic compounds. So actually to do the hazard characterization and combine that with exposure assessment and then finally do the risk assessments. So in this whole tox screening program, you also want to identify parameters and biomarkers that you can use for monitoring in humans, because we assume that the compound, toxic compound, or food ingredient, or pharmaceutical or chemical compound will be exposed to humans, and we want to be sure that we have a safe dose for humans. But as said, the way of doing this risk assessment, Bas calls it classical risk assessment, is more or less trying to do that for a general public. But of course, we have different vulnerable populations as well, like children, like women, pregnant women, or elderly. And we have all kinds of characteristics like sex and age, but also disease states that we have to take into account, but which we actually are not doing or not able to do that if we do, for example, animal experiments. But I will come back to that later on, because I think for the future risk assessment, it is important to include these different type of populations. And I'm also try to show you what kind of developments there are to take this difference into account. But in general, the risk assessment that it is now is mainly based on toxicological endpoints. So we look for acute effects, but also for more long-term effects. So in general, a single dose and repeated dose toxicity testing is performed in animals, rodents, usually rats, 28 days or 90 days exposure studies. We look for genotoxicity and carcinogenicity as a very important adverse effect, reproductive and developmental toxicity, but also local tolerance, for example, or allergic reactions, which only appear after repeated dosing. And of course, we were talking about human toxicity, but we also have to take into account the environment, because compounds would also reach the environment, and we want to be sure that the compounds are also not toxic for example, fishes in the water or any other organism. So how are these adverse effects detected? As said, many studies, and I have to say the regulatory test has been done with animals, rodents and non-rodents, but mostly rodents. But there are also other possibilities. So we can make use of in silico models, computer models like QSARs, PBK models, but also all type of different in vitro models. Cell lines are being used for decades now, but more recently also 3D tissue models, primary cells, stem cells, etc. are being used. And also ex vivo material can be used, so ex vivo material from humans, but also from animals, where you can make slices out of it, or can use the organs as such, as you see here in the photo, where you see a liver of the rat being used in a test system. And on the other side, on the in vivo part, besides animals, experimental animals, of course, it is sometimes also possible to have a look at what is happening in healthy volunteers, in patients, in occupational settings, etc. And I come back to that later, but what we want to do is to make more use of this human data, because the risk assessment in general is done to estimate whether there could be adverse effects in humans. So the more we know about the mechanism of actions in humans, the better we can predict the potential harmful effects. So to briefly touch upon animal experiments in the EU, in the European Union. So we have the we call it famous Directive 2010/33, which is about the protection of animals used for scientific purpose, where it is saying you are not allowed to use animals unless. So, and that principle that has, of course, influence on your choice of methods. So if there is an alternative, an accepted and recognized alternative, you have to use that. So you are not allowed to use them, animals. And for example, for specific applications like cosmetics, it's in Europe not allowed to use animals. And if you have to perform an animal test, of course, you have to take care and have to dive in some, let's say, setup of the study where you want to use the minimum number of animals with the lowest capacity of experience pain and suffering, of course. In general, roughly 8 million animals are being used in the EU, which is now without the UK, which is the country that stepped out of the European Union. And most of the animals are being used for basic research and applied and translational research. And around 20% of all the animals being used are for a regulatory purpose. So that's also good to keep in mind. And worldwide, we can only guess how many animals are being used, but the estimations are around a factor of 10 higher, so 80 million animals per year. In the legislation, it is clearly said that the 3R principle should be applied. So the refinement, reduction, and replacement of animal testing, if possible. Refinement means that you try to, for example, enrich the environment where the experimental animals are living. And also, of course, to use painkillers if needed. Reduction means that you can have a better look at the statistics, how many animals per group, but also make use, for example, of imaging, so that you can follow an animal during the life course and that you don't have to sacrifice the animals at all the different time points. And we can also, of course, have a look at replacements. So not using animals at all, but make use of in silico or in vitro models. So I would like to go with you a bit more further on the way of the alternatives. And I personally don't prefer the use alternative to animal testing because that's reference to animal testing. And I think we need to develop methods and include methods in the risk assessment that are human-based. And the human should be the gold standard. And I will come back to that soon. But why do we need alternatives to animal testing? And there are several reasons that express the limitations of the animal experiments. Of course, I think the very important reason is the scientific reason. So the limited predictive value for humans. There are more and more examples of animal models that do not predict the human situation well. And of course, it is also very difficult to characterize exactly the mechanism of a compound that causes a biological adverse effect in an animal, because it's more or less a black box. But on the other hand, there are also societal and ethical reasons. So there is more and more public concern about the use of experimental animals. So some people find it really unacceptable that animals are being used for our own welfare. So that increased the pressure on governmental bodies, industry, but also on the academia to find new solutions. And there is also there are also economic reasons, because doing animal experiments is very time and cost consuming. It's laborious. It takes a lot of test compounds, and also for more and more people of more and more industry, they find it not really fitting in the corporate social responsibility, and want to get rid of animal testing. And as I said, there is also sometimes legal requirements that forces to find new ways of doing the risk assessment. And what kind of new ideas, new approaches we have to do risk assessment. And also ILSI had some task forces dealing with this and look for a more holistic approach instead of have a look at the individual toxicological endpoints and try to define and save those from those single points, toxicological endpoints. So workshop was held in 2018, so that was after the publication of the paper Bas was referring to, with also a combination of academia, industry, and public organizations. And from that workshop, 2 papers were being published. And I would like to summarize this paper and share with you the main conclusions, because the title of these papers were "What are we waiting for?" And I think that's a very nice sentence, and also to keep in mind during the rest of the presentation. So if we look at the first part of these, the first of the 2 papers we presented, published, it is saying that there is potential for research strategies that reduce or avoid the use of animals by using more human relevant data from multiple sources, because we also know and realize that it's not easy and even not possible to rebase an animal experiment with one single assay. There is also discordance in the current legislation. So on one hand, it's promoting non-animal strategies, like in the REACH legislation and the cosmetic directives. But on the other hand, there is also guidance lagging, and there is not sufficient guidance how to make use of these non-animal testing strategies. So we should help companies, government, and academia to take steps in these directions, and therefore we also organize this workshop. And yeah, as I said, we need scientific developments and also strategies for the acceptability of these new approaches. So that was the conclusion of the first part. In the second part, we dive more into nutritional research, and also realize that there is a long tradition of using experimental in vitro studies, but at the same time we also do the in vivo verification with an animal model, while finally we have to say something about the human situation. But that's a step in between, by making use of animals, is often being performed. But from a regulatory perspective, there's no specific requirement for animal experimentation. So there's a requirement to provide information, and that is translated often into an animal experimentation, but that's not necessarily needed for regulatory perspective. But it is still, as said, considered as the standard, and yeah, guidance for alternative approaches is lacking, and therefore we have to build evidence and offer also possibilities for validations, new type of in vitro models, to ensure that these models can be used for their purpose. So to find those substantiating the dose levels and also to find safe doses for clinical studies. So I briefly explained how we do the risk assessment nowadays, mainly based on experimental animals, how we apply the 3R principle. And now I want to show you how we wanted to move from the 3Rs to animal-free innovations, where we want to have the human as the golden standard, make use of more human-relevant models, and also make use of all available existing data. There are data available, and previously they were published, or even not published, but put into dossiers that are submitted to regulatory authorities, but that was all paperwork. And but nowadays we have new models and computer systems that can help us to read this paperwork, and in that way we can try to make use of existing data. To show you that, to give you an idea about animal-free models that are being used in the past and also nowadays. On the right hand, you see some in vitro models, which I actually used by myself during my PhD. Sorry? Oh. There was someone not unmuted. Or there's someone. Can you mute yourself? Do you also hear this? Someone speaking?
Yes,
or
yes, someone is talking. Could you please mute yourself? I'm going to mute everyone, so that way I think it's better now.
Yeah, thank you.
Yes. No, we continue hearing the background noise.
Hugo is not muted. Hugo. Carry on.
I think it's better now.
Okay, thank you so much. What I was explaining on the right side, you see two models that are representing the human gastrointestinal tract. The upper part is being the large intestinal, and the lower photo is on the stomach and the human conditions in the GI tract. The reason why this type of model has GI tract are very different in rodents compared to humans. And we also know that the conditions in the GI tract has a big influence on the metabolism of compounds and the potential toxicological effects. So this was a first step to at least dive into the digestion and the metabolism in the GI tract that was more human-based than, of course, the rodent models. And samples that are being taken of this model can be measured for chemical composition, for example, and those outcomes, those concentrations of compounds can be used in, for example, PBPK modeling, and see how much of the compound is being distributed along the GI tract and, of course, the whole body. And then on the left side, you see new, more advanced in vitro models than we had in the past. Organoids, for example, or organs on a chip, where we can measure the effects of these metabolites from the GI tract on, for example, an intestinal organoid. And on this way, we can make use of human-like, human-simulated models instead of making use of animal models. And of course, there are advantages of of using in vitro systems, the interpretation of the biological mechanisms is easier. There are better controlled conditions, a larger experimental flexibility, and of course you also need less compound than when you have to feed all these animals for a very long time. But on the other side, there are also disadvantages because there is no interaction between organs. So most of the time, effects are being measured on a single organ or a single cell. And of course, in the human body, we have a combination of organs. So the biokinetics, the biotransformation, and is lacking. And also, there is no feedback mechanisms, for example, by hormones or all kind of immune parameters, etc. So, and also the chemical property of a compound can change in such an in vitro model, and we still have to make the extrapolation from a single effect in an organ to a whole organism. So still work to do. And that brings me to a very recently topic that is called the NAMs, the New Approach Methodologies, that are non-animal based that can be used to provide information in the context of chemical hazard and risk assessments. So actually, what we are talking about, it can be in silico approaches, but also in vitro assays, and it has, as long as it gives you information about the exposure or the hazard assessment. And there is a lot of discussion now going on what is NAMs exactly, and in my opinion, for me, it's not so important to define exactly what a NAM is and what it isn't. But it is good for you to know that we try to, this terminology is being used to summarize all the different type of methods there are available that are not based on animals, but are being used for human risk assessment. And in these NAMs, more and more new technologies are being used, other technologies than we knew from the past, from in vitro systems, that can be used. And for example, 3D printing is one of these new technologies developed for a very different purpose, but now being used in biology and also in toxicology. A lot of investigations on the microbiome, for example, but also microdosing as an possibility to investigate the metabolism of a compound dosed in humans at a very, very low dose, so that you can use it in a very early stage. So all kind of interesting new developments. And don't forget data integration and data science is a very important development, which is going faster as we could have expected a couple of years ago. And we try to make use of that to accelerate the transition to animal-free innovation. So human cell culturing is really booming. Where we started with the 2D cell lines, we are now making use of primary cells, try to combine different cell types, as you see on the above, the connection between chambers where different cell types are being cultured, 3D printed scaffolds where you can where cells can grow on in a more natural way. Organoids, as I just explained, here you see the villi of the intestine, lab on the chips, organs on the chips, and maybe also in the future where we a human on a chip, if we are able to combine different organs on one chip. So very interesting developments, and not all in the face of being validated and accepted, but more and more being used by researchers. So how can we move forward, and how can we make that real the transition to really prevent the use of animals? And therefore we need to integrate different NAMs that are based on human biology, as I explained. Try to combine it with adverse outcome pathways, so the biological mechanisms in the body, which Mathieu will explain you in the next webinar in more detail. The same for making use of PBK modeling, that will be explained by Nienke. And if we are able to combine these in vitro and in silico model with existing data, which we can retrieve from the public domain by using AI and large language models, and make use of the advantages of data science, we might be able to predict the different networks and try to predict what's happening in the human body. And of course, we have to think about validation, implementation, and acceptance. I won't go into detail, but in the past, we usually validated these type of methods in comparison with animal tests. And if we don't want to compare it with animals, because we want to develop something that is better, we have to compare it with humans. But how are we going to do that if we don't have all the data of effects in humans available? So that's something to keep in mind. And a couple of years ago in the Netherlands, we started with a project which is called the Virtual Human Platform for Safety Assessment, where we try to combine what I was just explaining. So trying to build a human platform where that is fed with data, all the data around, data on the physiological and biological mechanisms, and that is fed by 3 different human relevant scenarios. So case studies where we take into account, for example, the disease states, or the lifetime exposure, or sex and age. And in this way, we combine newly generated data and data that are already existing, and then try to predict what the effect could be on different humans. And the project, and we don't go into detail in the different work packages, that's not important, but what I want to explain, that this is the first project where we have toxicologists, data scientists, and social scientists in the project together. So it's not only the toxicologists, they are only 1/3 of the whole consortium, but all the other different disciplines are really needed. So toxicology itself is already an interdisciplinary domain, but that will be enlarged. That is already being enlarged with people from the data science and also from the social science. And that last part is very important if we want this to have these new technologies being accepted and implemented in legislation. We also have to take care, how do we change the rules? How do we change the way of working? How do we educate and train new professionals? So that's very important. And that brings me to one of my last slides. This interdisciplinary way of working, as I said, toxicologists, biologists, but also people that are very experienced in doing the right communication to the right stakeholders. Stakeholder engagement. So The project I just described is not being developed only by scientists. It's a co-creation of scientists together with industry, regulators, clinicians, but also consumers, NGOs, etc. And that makes this type of projects very difficult to perform, but I think it's needed to develop these non-animal strategies. Education, as being said, and we also think that this way of working might not completely fit in the current regulatory frameworks. So it might be that we also have to develop new regulatory frameworks. And to come back to that last sentence about regulatory framework, we thought maybe it's good that we try to move from prediction to prevention, because that's what we want to do. We want to prevent adverse outcomes and negative effects of compounds, instead of trying to predict what's not there. Because now the current risk assessment is based on trying to demonstrate that something is not toxic, but that's very difficult to prove. So we thought let's think about what is important to if you are have a look at if you have to do the risk assessment. We said compounds are considered to be safe if they do not have an adverse effect on healthy aging, both in a physical and a mentally state. So if we are able to prevent chronic diseases, if we can, if our vital functions of the organs are being present at all times, so that are not disturbed, the main functions of our body, that reproduction is as it should be, proceeded with normal fetal development, and that he can save, that he can have a safe life, and that means also doing your work safely, but also recreation, and of course, last but not least, it shouldn't harm the environment. And that was my last slide. And I want to thank you for your attention. And I couldn't see you, so I couldn't see whether there is a question or not. But I tried to stop sharing.
Okay.
This.
Thanks a lot, Cyrille. This was a nice overview of where we are at the moment. And we need to carry on from here. Also, for the sake of our human, let's say, well-being, I propose that we have a 5 minutes break now. In the meantime, if you have questions, please put them on the chat. I have seen a number of questions on the chat, but they were mostly on the possibility to hear you properly. So if you have, let's say, questions about the contents of our lectures, please let us know.
Yes, I'm happy to answer your questions, and apologize if you don't hear me all the time. But please feel free to ask, and I'm happy to answer by chat.
Yeah, okay. So in 5 minutes' time, we will be back. Ja, Cyrille, ik had grote moeite om in contact te komen zojuist. Zelfs vanuit de universiteit.
Ja,
Mijn camera doet het hier op de een of andere manier niet.
en ik was blijkbaar ook moeilijk te verstaan.
Zo af en toe viel je weg, ja. Ja.
Ja, oké. Maar goed. Ja, het is niet anders. Ik heb mijn best gedaan.
Ja, zeker. Ja, ja. Mooi overzicht.
Ja, volgens mij was het wel oké, toch?
Zeker. Ja.
Of was het te lang?
Helemaal niet. Nee.
Oké. Was it okay, Geraldine? Did you, besides that it might be a bit... Oh, okay, it's fine. You take over from here, Bas.
Yes, I do.
There were moments, Cyril, where the sound was a bit not stable, but in others it was okay. But at the end, it was okay.
Okay, great.
We already have some questions on the chat.
Yeah, I will have a look at it.
Right. Share slides. Okay, can you see my slides?
Yes, all good.
Yes.
Okay, thank you very much. So, to conclude today's session, I would like to dive a little bit deeper in understanding what's happening on the cell level, because one of the issues that is important to realize is that apart from all the things that we need in an integrated approach, as explained by Cyrille, it is essential that we understand the fact that the toxicity of a compound, and this is just a reminder for you, is determined by the interaction of a chemical with the biological system, resulting in an adverse effect. And since all these interactions, or nearly all these interactions, are taking place at the cellular level, we can have a lot of information coming out of in vitro systems in what we now call the field of in vitro toxicology. And this is only possible because over the last 3 or 4 decades, we have an enormous development in the field of cell biology. We understand much better what's happening in the cells. So we also understand much better what can go wrong in cells and what is the meaning of this in terms of, let's say, adverse reactions of a compound, of a chemical. You see here all the things that are in development. We have the general cellular biology is going on. We understand much better what's going on in the field of differentiation, apoptosis, as one of the principles. And mind you, if we talk about apoptosis, I come back to that later, and talk about necrosis and apoptosis. Mind you, apoptosis is a normal biological process that we see in the bodies as a essential part of the development of an organism. So if we follow this list, we know much more about cell-cell interactions. And this is also one thing that also Cyrille was mentioning, that we can work with isolated cells but it is sometimes essential that we know more about, let's say, the interactions of cells and what is the relationship there. When I said that, let's say, most of the interactions are on the cellular level, most of the follow-up steps of this reaction have to do with cell-cell interactions in an organ. We also know much more about signaling processes, for instance, in the field of receptor-mediated processes. We know, of course, a lot of things in the field of genomics, proteomics, metabolomics, or metabonomics, or whatever you want to call it. And all these things can be headed under the process of systems biology. So the field of in vitro toxicology, which is, well, more or less, well, what is it, 35 years old now, is developing because we can study a lot of things in vitro systems, and that gives you at least the intrinsic ability of a chemical to give a certain toxic action. That's one of the things that is important to mention here. Okay, the joke in between. What is in vitro? What does it mean? We could make it a quiz question. Well, most of the people don't understand that in vitro means in glass. And well, so this is an in vitro experiment, you see. You see the mouse in the glass. It drank all the whiskey in the glass. You can see it on the eyes of the animal. So is this an in vitro experiment? Maybe. But actually what we see nowadays, it's much more this sort of thing. We see plastic plates. So do we have to talk about in plastico? Right. I see a number of chats going on. Can you have a look at it, Cyrille? Maybe they are for you.
No, just joking. They are laughing about
Okay.
plastico.
Good. I cannot see them. Yeah. Okay. Well, let's talk about, let's say, cytotoxicity in more detail here. There are things that are influencing each and every cell type. And that's what we call basal cytotoxicity. And this is related to the household functions of each and every cell. And this has to do, for instance, with the integrity of the cellular membranes. Not only the outer membranes, but also the ones inside the cell. So it also, we have to talk about, let's say, the maintenance of the cellular compartmentalization. Is the nucleus intact? Are the mitochondria intact? Is the endoplasmic reticulum intact? Etc., etc. Because that defines the well-being of the whole cell. And another thing is that we see is that we have to have an intact energy supply mechanism. For instance, in the mitochondria, is the production of ATP in order, etc., etc. Those are the things that are influencing each and every cell. And what we see, for instance, is that if you look at the things that are happening in an acute toxicity system, for instance, in animals, if you are more or less killing the animal, it is because you are killing the cells. And this is most of the time related to the basal cytotoxicity of it. But it's not all. It's not all. We have also the differentiated functions of cells. In each and every organ, you have things, and that can be defined as, let's say, cell-specific functional disturbances. If this is taken into account in the toxic action of a compound. I give just a few examples because the list is endless. And we have to realize this also if we are developing in vitro methodologies to measure this. I come back to that in one of my other presentations. I just mentioned the neuronal activity. If we don't have neuronal activity, the organism cannot work. So if we have a specific compounds that are neurotoxic, we have to take that into account. If we are looking at, for instance, the liver functions, one of them is the production of albumin. That's an effect that you can see on the hepatocytes, but it influences the rest of the body in an enormous way. Because if you stop the production of albumin, or if you stop the excretion of albumin from the cells into the rest of the body and via the blood, then you have a problem as well. And last but not least, I mention, let's say, specific barrier functions, specific transport functions as an essential element of many parts of toxicity studies. So when I said the whole thing starts by influencing cells, it can be on the basal cytotoxicity, it can be also on more cell-specific functions. When we are looking at, let's say, the mechanism of action and how this can be translated in adverse outcome pathways, we have to take into account the fact that these are the starting points, the molecular initiating events are, as they are called now, but we need to take into account what are the next steps as well. So here you see a cell that can be influenced on many ways. We can have effects on the membrane, for instance, by the influence of reactive oxygen species. We can have effects on the DNA. We can have effects on the mitochondria, etc., effects on transporters in the cell, all kinds of things that are easily studied in vitro and that need to be placed in the proper context of what it means for the organism, for the organ and for the organism. We now have a long list of possibilities to take into account. Oxidative stress, general cell responses, ER stress, for instance, induction of metabolic enzymes, metal binding proteins, for instance, whole things that can be taken into account as so-called biomarkers for cellular effect. And this list is also very long. Having said this, it means that we have to take into account also another very important issue of toxicology, and that's the fact that we need to know more about the possibility of a compound to react somewhere in these processes. So if we go to that scheme that we presented in this paper in 2016, you will see that there are steps in the whole process that are more or less decision points on what to do. And some of these decision points are driven by the fact that we know much more about the structure of the compound via quantitative structure-activity relationships, for instance, that point you to the interest in certain biological processes on this long list of biomarkers. So, if we are studying general cytotoxicity, we have a number of methodologies to do this. You can stain cells to make the difference between live cells and dead cells, for instance. You can see how cells are functioning by measuring their metabolic mitochondrial capacity. And one of the tests that are used is the MTT test. We have tests for oxidative stress, and we have this very long list of functional endpoints that also can be taken into account, driven by, on the one hand, the structure properties of the compound, but also driven by the possibility for the compound to reach these endpoints. So here kinetics also play a role, and we will come back to that in the presentation later in Nienke's presentation on Thursday. Just to give you an example of such a test. And by the way, this is a test, an in vitro test, one of the first in vitro tests that was recognized by the OECD as a validated test. What you see here is a phototoxicity test. You see here rows of, I don't know if the pointer works. Does it work? You see rows of cells that are given a certain concentration of a compound. And if you go from left to right, you can see that the concentration, if the concentration goes up, the color is changing. What's happening here is that after the experiment, the cells are stained with a neutral red that shows that when the cells are alive. So only live cells are taking up neutral red in their cells. And so you can stain them afterwards. And what you see here is that on the higher concentration of the cells are dead. And this is a control again, so to show that there is no effect from one well to the other. Oops. On the back side, on the lower part of this figure, you see the same plate, but here the plate was placed into UV light. You can see here that the concentration of cells that are producing the toxicity are much lower than in the dark situation. And this shows that this compound, in principle, has the possibility to be phototoxic. Now, actually, we know a lot about the possibility of a compound to be phototoxic because we know that light can more or less change the structure of a chemical, and we know a lot about which chemicals can do this and which don't, because this is given by the structure of the compound. And what's then happening is that if these cells are illuminated with UV light, for instance, they can produce reactive oxygen species. And they can then, depending on the concentration, have no effect because the threshold was not surpassed. They can give apoptosis of cells, or even at the highest concentration, necrosis of cells. So since we know a lot about these mechanisms, we can predict on the basis of this cell system the possibility that this compound is phototoxic. Mind you, we are talking now about, let's say, the intrinsic ability of a compound to do so. What we have to do also is to know if these compounds can actually reach the skin. If you swallow it, a compound, and the kinetics of the compound show you that there is hardly any exposure of the skin, then it is in vivo not phototoxic. So you have to be aware of the fact that such an in vitro system can give you a number of false positives if you don't take into account the kinetics of the compound. So how do we interpret these results? What does it tell us about the in vivo effects? And one of the things is that we have to take into account the dose, and we have to take into account the kinetics of the compound. And these are the subject of the lectures that we are giving on Thursday. So, without further ado, I think I would like to end the presentation by taking your questions, but also to refer you to the webinars of Thursday. And I really hope that you found these presentations as a general introduction in what we are talking about as a good one, and that you are warmed up for the things that are happening on Thursday, and even more so that you will be able to join us for this hands-on workshop in which we are going to try to use the scheme later on in July. Having said this, I now take questions, and thank you very much for your attention. And I will stop the presentation here by going back to this. There is a question. I'll read it. What I would like to know can be responded later on. Oh, yeah. At the current state, how can we assess the disruption of epigenetics caused by toxic products in different cell types? Are animal models the right ones? What is the perspective developments? Yeah. So epigenetics is one of the issues that is mentioned here in the question. Tanguy, thank you very much for this question. There is a possibility to take these epigenetic things into account, and probably we will come back to that later when we are talking about adverse pathways observations, because that is a problem. We know that, and it is, you know, quite sure that we have to take that into account. Cyrille, can you add to this, maybe?
Yes, we have to take that into account, but we are also still do not know how to do that exactly. So yeah, that's something I'm not aware of. Many new developments in that field. So the recognition of the importance of it, yes. Animal models I'm not aware of, that taking that definitely into account. Maybe you, Bas, you have heard something?
Well, I know that there are people working with organoids, in which they are following them up over a longer time, and where, let's say, these epigenetic problems are also taken into account in the development of these organoids. But it is in its infantic situation, more or less.
Yeah, I agree.
Yeah.
So it is possible to investigate it, but there is no, let's say, model being developed or assay. I have to say assay being developed that takes this into account.
No, that's true. It's in this infantic situation.
Yeah.
We realize that it is there, and we realize that it is an important issue in many cases. It also depends on the question that you are asking. If you are talking about, let's say, compounds that are having a very long exposure, the epigenetics are probably more relevant than shorter exposures. But we know examples of human exposures of epigenetic things, like let's say hormonal issues, the hormonal disruption of things is in some cases related to epigenetic things. So we can try to model that also in vitro systems, but it's very difficult to do that. Okay, we think about this and we come back probably if there are answers. We come back on that on Wednesday, on Thursday. Are there other questions? What I would be interested in is we now have something like 40 people in the group. It would be good to think about the possibility to join us in the July meeting. We don't have many registered people there at the moment. Geraldine, maybe you can give an answer to that. And we are still hoping that, let's say, in the next week or so, we can have more people that can join us. We have a deadline for that in the
7th of June. I can give a follow-up on that. It's like for the in-person training, we need to be a maximum five persons per people, and the training is based in Belgium here at ILSI Europe offices. So if you are interested in this in-person training, I invite you to please register in the link I just shared on the chat. And we have the deadline for to register is until the 7th of June.
Okay. In the meantime, yes. I see that there is another question from Mari. Complex mixtures. Actually, we will come back on this in the presentation at the end of the Thursday presentation. I will point to that.
Yeah, and to briefly answer, or maybe not to answer it completely, but it depends really on what kind of mixture it is. Are there compounds with the same mechanism of action or not? If they have different mechanism of actions, you can also imagine that you need different type of cytotoxicity assays. So you need to know something about the compounds to be sure that you choose the right cytotoxicity assay. And some of these compounds interact with the readouts of cytotoxicity assays. And that's also important to keep in mind. So there is definitely not one assay you should use or can use, but that may differ case by case.
Yep. Okay. Well, if you have more questions, please don't hesitate to contact us. We are approaching the time of 4 o'clock now. So I propose, Geraldine, that we keep the line open for people that want to ask questions until 4 o'clock.
Yes, no problem on that. And now we can't hear you, Bas. There's
audio.
We hear you, but very low.
Okay, is it better now?
No. It's like you are not close to your microphone or something.
I didn't change my position.
Much
now
better.
it's better. Now much better. Yes.
Okay. As we said, we keep the lines open until 4 o'clock. And if you have more questions or remarks to be made, positive or negative about our lectures. We are very, they are very welcome. We also can learn, long life learning, isn't it? And we will see you then again on Thursday, and I really hope that my camera is working then.
We hope the same.
Yeah. So thank you very much, everybody, especially Cyrille, Geraldine, and Hugo for the support, and Cyrille, of course, for the lectures. And
Yes, thanks.
thank you very much.
And just a reminder, and if you want to know more about ILSI Europe, I'm going to share also the ILSI Europe's website link. Just a second. So there you can see all the activities ILSI is developing, and also the different task force we have, including the alternatives to animal task force.
Yeah.
Animal testing.
We had, by the way, Cyrille, we had a discussion in the board of directors not so long ago about the name of that work, of that task force. Should we change it to NAMS?
Fine for me as well.
Good.
So there you have the ILSI's website, and you can go there and give a look and see the activities we are developing right now, and also the ones coming.
Right.
I think no one has questions, so I think, Bas, we can close today's webinar.
Right. Well, thanks again for everybody that was joining us today, and we hope to see you back on Thursday. Thank you very much.
Yes, thanks a lot.
Thank you.
Thank you. See you on Thursday.